It's a sign of my advancing age that this is the point where I realise I've forgotten my reading glasses. Try to manage. Okay, so my name's David Silver, and I'm acting Dean of Science, I hope fairly temporarily, and as such it's a real pleasure to welcome you to the inaugural lecture from Professor James Russell. And also welcome you to probably the strangest lecture theatre in the university. A weird place to be. So James has been here, man and boy, since 1997, apart from 2 years doing research on French tropical islands. So it's a sort of a message about how you choose your research areas, where can it take you to do the research, and that was obviously a good choice. James is a prolific and influential researcher in invasive species management with nearly 200 publications, many of those in very high impact ecology journals, and more than 5,000 citations. And he's got a list of prizes that I'll try to get right. He's been awarded the Prime Minister's Emerging Scientist Prize, the University of Auckland Early Career Research Excellence Award, University of Auckland Not Early Career Research Excellence Medal, the Society for Conservation Biology Distinguished Service Award, and a Rutherford Discovery Fellowship. So it's an amazing string of honours. But James tells me that the thing he's probably most famous for is for losing a rat and being unable to catch it during his PhD, an incident that he was able to turn into a nature paper, the front page of the New York Times, and a children's book by Winnie Ihimara. So he's now starting to feel his age. And the children who first read that book are now our first year students. So just wait until you hit the point where you're now teaching the children of the first year students. James's teaching has included teaching field work methods to influence undergraduate students who are, because of the city we live in, increasingly disconnected from nature, so it's a great thing to do. Emphasising how important it is that graduate students explore the philosophy and ethics of their science practice as well as the science itself, and combining his parallel expertise and passion in both ecology and statistics. There's a great deal of service, both nationally and internationally, in James's record. He's worked closely with DOC to provide a scientific basis for policy and management, showcasing New Zealand's island conservation expertise to the world, and of course there's a leading figure in predator-free New Zealand. James and I do have 2 things in common. I've been a stoke volunteer for quite a lot of years, there's some other people here, and there are 15 rank boat stations around my house just now, so that the content of James's lecture is, I think, of particular interest, not only to me, but I bet a lot of us really share that this is important to our lives. And I also think that we're the only new professors here to show pictures of Swiss Army with like knives in our inaugural lecture slides. So please join me in welcoming to the stage Professor James Russell. The floor is yours.
Thank you very much, Doug. And it's, I must say, thank you all of you. It's wonderful to see so many friendly faces here this evening to be able to celebrate this occasion with me and hopefully tolerate listening to me for about an hour as I go through really what's been a career's work so far that's led me to get here. And what I want to emphasize today, tonight, and communicate to you is how in trying to be a conservation biologist, you really have to escape those disciplinary boundaries where you think this is the tool I want to use and apply to every problem and go, what is the problem and what tool do I need to draw upon to use it? So like the Swiss Army knife that my mother and father gave me when I was young and I took hiking, tramping all the time, and there was always a tool for every occasion. That's the approach I've come to realize is necessary for conservation biology. And that Swiss Army pocket knife ends up being quite a powerful tool, but one where you're always wondering, where's that tool that I need right now? Or what's this tool actually supposed to do? But I think I'll try and use it anyway, even if this isn't exactly the way I should be using it. So I'm going to start. This is me when I was young and clearly a fan of Sesame Street. But also a fan of birds. My passion as a New Zealander is for birds. Ironically, these photos are from Wilson's Promontory in the south of Australia, and these birds, although used to be found in New Zealand as an introduced species, which I suppose is also ironic given that's where my career is now focused. We're a very friendly pet that you could find at Wilson's Promontory, and they really help me engage with nature. And as Doug alluded to, it's absolutely critical, I think, that people engage with nature in a very hands-on way, and we create every opportunity possible for them to do it. So it's fair to say I like our feathered friends quite a bit and want to do everything I can to protect them. So naturally, and particularly as a University of Auckland undergraduate in the 1990s, all paths led to Tiritiri Mātangi Island, which John Craig and Neil Mitchell set up as a groundbreaking, pioneering project for not only New Zealand conservation, but global conservation. This idea that you can take a relatively heavily impacted site, in this case a farmed lighthouse island, and restore it into something today where if you go out to Tiritiri, it seems like it's the original forest and the original birds have all been placed back there. And I had the opportunity to go out there in 1991, myself there in my, clearly my favorite skateboard jumper with my little brother Douglas, who's doing a PhD in psychology in Melbourne at the moment, and my father there who was forced, as he said, I led my brother off on an adventure around Tiritiri to look for some birds, and my father spent about an hour wandering around the 200-hectare island looking for us. This was for the Tākehē release on Tiritiri Mātangi, and it was absolutely amazing to see a bird such as that, presumed extinct, rediscovered in the 1930s in the Murchison Mountains, brought back to Tiritiri Island, and really amazing to be there for it. And I did love the Tākehē as a child. So when I came to the University of Auckland, I wanted to be a bird conservation biologist. Due to some twists of fate that many of you are probably aware of, the only PhD that Professor McClout was offering at the time was on rats. So put aside your love of birds, because for the rest of this hour you're probably going to hear a lot more about rats, which are the primary threat to our native birds of New Zealand. Fast forward to 2010, when I start a faculty position at the University of Auckland. This is Greg the Tākehē on Tiritiri Mātangi, perhaps remembering me from years past. And I'm not sure, again, it's hard to tell whether he's liking me or hating me there with what's going on. But certainly we had another personal engagement that once again just emphasizes how much I think having those interactions with wildlife are so critical for people as we want to help them engage with conservation of nature. So conservation biology, this topic that I've apparently dedicated my life to, it's the study of the conservation of nature and biodiversity with the intent of protecting species, their habitats and ecosystems from excessive rates of extinction. As I've learned recently from working with colleagues in philosophy, there's quite a lot to unpack in just simply saying we want to conserve nature and we want to conserve biodiversity. So at the end of this talk, we're going to come full circle from the biologist engaging in the philosophy that was ostensibly the subject of a doctorate in philosophy, and wondering what is nature and what is biodiversity and what do we really want to achieve at the end of this with our time. I'm entirely motivated by what you'd call translational in the life sciences, or action-based in the social sciences research. If my research isn't changing what managers do and what people do and what we do as a society, then most of you are probably aware I become demotivated pretty quickly about the nature of the topic. But what's absolutely critical is that New Zealand is uniquely placed to lead in island conservation. We are one of the few island nations in the world that is an affluent society inhabiting an independent island state. So that is a powerful combination of two things coming together, which means socially, politically and culturally, it's not a coincidence that we lead the world in island conservation. It comes about specifically because of those circumstances. The other places in the world where I've worked which come close to this are Reunion Island, a French island off the coast of Madagascar, but it's as much part of France as Hawaii is part of the United States, another place I've worked, where you have two affluent nations living on an island. But both of those island states are burdened by the fact that the predominant mindset is from continental ecologists. And what you'll see as the recurring theme tonight is that in continental ecology, it's all about mammals and predators being a natural part of ecosystems. But that's not the case on islands. And so very early on in its history, New Zealand deshackled itself from those continental mindsets that impinge on conservation and took a purely island-based approach. So I'm very proud to continue being the torchbearer for the importance of that approach. Yet, as much as New Zealand's unique in that respect, the biodiversity challenges New Zealand faces are common to every island on the planet. We're not particularly special in any regard. We're just like the Galapagos, or the high Arctic islands, or the islands of the Western Indian Ocean, or indeed any island where people brought introduced mammals with them, and the impact of those introduced mammals were devastating. So what we do in New Zealand can be translated entirely directly to the rest of the world. And this is shown here by this analysis we undertook a few years ago of threats to IUCN red-listed species. So the IUCN, the International Union for the Conservation of Nature, maintains a list of species that are critically endangered with extinction. What you can see here is in the orange are all of the red-listed species found around the world, and this is the ranking of the threats to them as of today. Overexploitation, agricultural activity, and urban development are leagues ahead as the major threats to our biodiversity today. But what's interesting is when you look in the blue bubbles, you see how many of those species are species found wholly on islands. And the most shocking statistic there, to be honest, is that just under half of the IUCN red-listed threatened species are found wholly on islands. Islands make up only 5% of land area, but they make up 50% of species threatened with extinction. And the other point I want to refer you to is that if you look at those statistics, it's the blue invasion and disease one, where 73% of species on islands threatened by invasion and disease are species threatened on islands. So by far, invasion and disease is the only threat more likely to be impacting a species on an island. So traditionally we think of invasive species as things like stoats and rats and possums, but now we're all learning that disease in the forms of kauri dieback, myrtle rust, or even the global pandemic of COVID-19 are the kinds of threats of humans introducing things well beyond natural boundaries and the impacts that follow from those. So it's no surprise then that as an island nation focusing on conservation, all paths naturally lead first to trying to solve the invasion and disease threat, and after that we can try to remediate some of these other equally important threats in that they pose a risk to species extinction. So far to date, my Swiss Army pocket knife of conservation biology tools, which I will go through one by one tonight as I flick it out on my metaphorical Swiss Army knife, is fieldwork, computer science, molecular ecology, biogeography, population biology, mathematics, community ecology, stable isotopes, statistics, social science, phylogeography, macroecology, and finally philosophy. So let's start with field ecology, where it all really began. The question here is, how do we detect single rats invading islands? This was the question Doc posed for my PhD topic, Doc funded for my PhD topic, and Doc strongly suggested I find islands without rats and reintroduce rats to them to find out how rats invade islands. I couldn't say no to a topic such as that. Thinking of all of the media headlines, I was bound to end up in one way or another, and indeed that bore out. The result of this study, although quite painstakingly for a 4-year PhD, was I managed to release a total of 5 Norway rats individually onto an island and radio track them for 4 weeks. The outcome that changed conservation practice was we had confidence in intercepting rats reinvading islands, thus clearing islands with high reinvasion rates. We can now tackle islands like Rangitoto and Motatapu. The attitude now is not that rats will get back there, but of course rats will get back there, but the question is what do we do when they get back? It's exactly the same with our COVID border biosecurity. We know COVID will cross into the border. How we are judged is by our response to it once it crosses over into the border. And so I'm very pleased that that body of field ecology work from my PhD created that change in policy and confidence for the Department of Conservation. So this is what happened. It meant basically going out to an island, and as I said, working my first 9 to 5 job. It was 9 at night till 5 in the morning, and every hour on the hour I would wander around this 10-hectare island in the Hauraki Gulf with this radio antenna and try to locate a single rat upon it. And that was quite exhilarating at times when there was lightning, but nonetheless data must be collected, so as a PhD student one takes those risks. We collected data points here of where this rat was located on the island every hour over 4 weeks. So the first rat, the second rat, That third rat's only about a half rat because he managed to wrestle his radio collar off him. And there's a fourth rat there. I'm not even going to show you a fifth rat because the collar failed. So actually I only ended up with 3.5 rats worth of data. But those are just details, really, in the history of time now. We did manage to get a paper out of this. I collaborated with a colleague who was doing his PhD in neuroscience, and he had a model, a simulation model for neurons firing around in brain cells. And I said, I think I can apply that to my rats bouncing around the island. So what we see here is on the top left there is the area of the island that a rat would cover in any single night. And it comes out just shy of 2 hectares. So a rat at its peak activity covers about 2 hectares. But what we see on the top right there is every day the rat explores a new part of the island, increasing its range across the whole island until after about 3 to 4 weeks it's explored the whole 10 hectares on the island. But the question then is the rat being systematic and having some deeper meaning to its exploration, or is this really just come about by chance? So that's where that random simulation model came into play. And what we found here was that the rats were, although there's some signal they might have been being systematic, really they're just doing what the computer scientists call a drunk man walk, which is wandering around the island and seeing where they end up because it's all very new to them, like when we explore a new city and just kind of find where we end up without having a map to follow. But as some of you, and stop me if you've heard this one before, this rat, the first rat, Razza, started here and did a little tricksy thing on me when his radio collar stopped working and ended up here. So obviously I think most of you know what happened next. What happened next was a bit of this on the island, which, by the way, is a genuine Swiss Army knife moment, because this was in the days when wine bottles were corked, and if you didn't have a Swiss Army knife in your fieldwork trip kit, you'd be in a bit of trouble. But then after that, this happened. And it was turned into a proof of concept that there is indeed at least one rat in the world that is very, very difficult to catch when it gets back to an island. So then you may be able to guess what happened next. There was a bit more of this, but in a good way. But then what happened next was even stranger, was unbeknownst to me, Whittier Hamida decided to write this. And I do have a copy of it here tonight if anyone wants to read it, but rest assured you don't have to, because as well as being on the front page of the New York Times, you can listen to it as a download on Radio New Zealand as a soothing bedtime story, 20-minute bedtime story to put you to sleep. So the next question was, how do we determine which species of rat is invading an island? And here, some tools from computer science proved really powerful. The method we used was pattern recognition and classification of rat footprint tracks. So we've got 3 different species of rat in New Zealand: Rattus rattus, Rattus norvegicus, and Rattus exulans. The black rat or ship rat, Norway rat or brown rat, and the Polynesian rat or kiori. And so when we find rat footprints going through a tracking tunnel, we don't know what species it is, and we need to tailor our biosecurity response to those different species. And what we found using these computer science algorithms is an 80% success rate assigning rat footprints to the species. And the outcome of this is we can tailor our biosecurity responses to the rat species, and now with further advances develop digital devices for rodent biosecurity. Instead of using tracking cards, we can now use the touch screens of cell phone devices, and the rat walks over those, and in real time you can determine what the species is and prepare your biosecurity response. So here we see, it seems quite, the eye is an absolutely powerful pattern recognition algorithm. We can see, or at least a number of you in this room will be able to see the rat footprints in here. I know many of you will have stared at hundreds of these cards, if not thousands in your lifetime. We see those rat footprints. It's very hard to convince a computer that the black dots make patterns of these rat footprints, but nonetheless we created an algorithm that could do that automatic pattern recognition. And then it could identify them and confidently assign them to this is a Norway rat, and then we can launch a Norway rat tailored response. So that was really powerful for the Department of Conservation as well. Then more questions came along, and towards the end of my PhD, the Department of Conservation wanted to know, how do we know if rats repopulating an island are survivors or reinvaders? You complete your rat eradication, those reinvaders come because you're doing riskier eradications, but then suddenly you've got rats across your island, and you don't know, are these ones that survived from the eradication a year ago or swam over very recently? And at this time, microsatellites were the cutting edge of molecular biology, and we used genetic fingerprinting of rats, and you can match them with either the pre-eradication or the potential source population to determine whether these are survivors or reinvaders. In this case, thankfully for DOC, we determined that their eradication methodology was still successful. They hadn't failed at the eradication, but the rats they had were very, very rapid reinvaders. This was for a small island off southern Stewart Island, where they did not expect rats would be willing to swim across the very cold channel, but nonetheless they had a dozen rats swim over within a year of that eradication being completed. The outcome of that changed management practice was that genetics is now a standard operating procedure for rodent eradications and biosecurity. And so those genetic samples are collected routinely and laboratories are analysing them. So to show you that in detail, this is Pearl Island off the southeast coast of Stewart Island. DOC were only monitoring the rats around the coast, and indeed they found all these rat captures here, so they weren't sure whether these were survivors around the coast or reinvaders swimming over. So here we can see what a plot looks like. Those blue dots on the upper left are the Stewart Island sample. So if a rat comes from that population, that blue population, we know it's swum over from Stewart Island. On the lower right-hand side, we see the pre-eradication, the original population on the island. So all we have to do is take new rats that we capture on the island and assign them to either the blue dots, which means they're a swimmer, or to the red dot, which means they're a survivor from the eradication. They had 2 species turn up, not just 1 but 2 species from this 3-species rodent eradication, and they all assigned to the Stewart Island population, which meant DOC knew with confidence using microsatellite genotyping that rats were swimming over very rapidly from Stewart Island, and biosecurity was the issue on this island, not eradication implementation. So with my time done in New Zealand, it was time to move to this gentleman's private atoll in French Polynesia. This is Marlon Brando, and this is Teteroa Atoll. He purchased it in 1967 when filming Mutiny on the Bounty. And being Marlo, being a little eccentric, he prevented any development happening on it. What he did do is in that island in the upper right, he developed an eco-hotel and invited all his Hollywood colleagues to come there. They weren't quite prepared for Marlon's interpretation of an eco-hotel, which was literally just a thatched room and a serve yourself at the bar and cook your own meals. So he never really got the big hotel going that he thought it might be. But the island was relatively pristine and protected as compared to most of the other atolls in French Polynesia during the 80s and 90s hotel boom. So I ended up finding myself in the tropics, which was a bit of a change, and I spent the next 2 years working in French tropical islands trying to implement some of the conservation tools we have from New Zealand for their very similar problems. And the first question that came up is, when is the best time to target rat population for eradication on tropical islands? And the method we used here was a biogeographical one of a global analysis of rat eradication success on islands. The result was we identified 4 different island biome types where eradications tend to happen around the world. And the outcome was that now rat eradication is tailored to the island biome, whether that be in the tropics or the temperate regions. So let me go into a little bit more detail on that. What we actually did was collected environmental data for all of the islands around the world where eradications have occurred. The red crosses are successful eradications, the black triangles are unsuccessful eradications. And you can see here we collected data on the variation in temperature across a year. So islands such as in temperate New Zealand can be quite warm during summer but quite cool over winter, whereas in the tropics, islands tend to have reasonably constant variation in temperature because they stay quite hot. We also have precipitation and rainfall, so you can have wet and dry seasons, or you can have constantly wet islands, and also on the amounts of rainfall and temperature themselves. So we managed to classify through a clustering process these 4 different island types. So you can see on the left-hand side of the temperate islands of New Zealand, a very high success rate for eradications, 90%. But then we have 3 other biomes, so not all tropical islands are created equal. We have savanna islands, very dry islands, where the success rate is very high for eradication. We have tropical seasonal, wet dry islands, where there have been a lot of eradications, but also a good number of failures. But the really concerning area were the tropical wet islands. They're constantly warm and they're constantly wet, which creates a dream habitat for rats and a failure rate for rat eradications around 50%. So by interpreting these 4 biomes, we ended up trying to work out when timing of eradications could best be done. So in temperate New Zealand, we see here that in winter things cool off, primary productivity dries up, and the rat population then lags. So that blue area is that lag when rats are vulnerable. It's happening right now around us once again. The rats are hungry, the rats are cold. Most of you are probably experiencing them coming into your houses in some form, and we're trying to remove them. So in New Zealand, we know you target eradications at winter. In contrast, on the top right, there is tropical seasonal forest. It tends to be quite wet and has a very short dry season, but that dry season is a great opportunity to target the rats. But that dry season isn't necessarily during summer or during winter, it's just when the dry season occurs in that part of the world. Down the bottom left, we have the savannas, which are mostly dry, but they have a rainfall pulse. That creates an explosion in the rat population, but just after that explosion, like a mass seeding event in New Zealand, the food dies off, the rats become hungry, so you target the rats just after that rainy season on those savanna islands. But once again, we end up at the very concerning tropical rainforest islands where it's wet all the time, and to be honest, there's not really a clear time to target them. You just have to target them with all guns blazing, recognizing that those rats are probably not looking for food because there's lots of alternative food, and they're probably breeding at the time, which are really challenging conditions for rat eradication. And as I learned, this was 10 years ago when I was only applying temperate experience. So myself and Sandra Anderson and Mike Lee tried to undertake one of the early rat eradications on tropical islands. And it turns out a really good time not to do a rat eradication is during a cyclone. That's a really bad time to do an eradication. So this is also a Swiss Army knife moment, because you're told you've got 1 hour to evacuate the island, and everything you take with you is all you will be taking with you. So in my hands there, I think I see my field notebook. I think my laptop's there, and my passport, and my Swiss Army knife, because we were promptly evacuated and dropped off in Papeete and told we'd been saved, even though all of our bags were left on the island and we had 24 hours until our flight. So then moving to the Western Indian Ocean and applying some population biology tools, do rat population dynamics change in coexistence with introduced mammals? Rats on an island by themselves are reasonably straightforward to eradicate. It becomes more interesting when those rats are competing with mice, or when those rats are coexisting with cats preying upon them. So what we did here was a capture restudy of rats on islands with different suites of introduced mammals. The result was rat population dynamics do differ substantially in the presence of coexisting mammals, and the outcome which changed management is rat eradication method and timing is tailored depending on the coexisting mammals. So off to Europa Island, the French maintain a number of military base islands around the coast of Madagascar. They're called the Scattered Islands, the Îles Éparses, and this is the largest of them. So comparing here in the north of the Mozambique Channel, Juan de Nova, where there were mice with ship rats with cats, and with Europa in the south, where ship rats exist alone. The question here becomes, how do rat population dynamics differ in these two islands? And what we found were, at the top you have the population dynamics of the rats on Europa, and at the bottom you have the population dynamics of rats on Juan de Nova. Key things to see here is when the rat exists alone, it very much feels it's living the island life. It's not competing with any other species. Whereas on Juan de Nova, it feels much like they're just back in downtown New York. There's cats, there's mice. It's a pretty busy time. So what we see here is that there's pulsed recruitment on Europa Island, so the rats are reasonably lazy and they don't necessarily breed over winter, they just breed over summer. So you do see the population cycle, but it's because of pulsed juvenile recruitment. So there's a big breeding event in summer and not so much of a breeding event over winter. You also see an elevated density. In the absence of cats and mice preying upon them and competing with them, the rats can maintain a higher density on Europa Island. And finally, you do see that stronger population cycling because of that pulsed juvenile recruitment, whereas on Juan de Nova, the population cycling is dampened and the rats on that island tend to maintain themselves at a lower density. So moving on back to Reunion Island, the headquarters for this research project, the question was, does cat control release rat populations, increasing impact on seabirds? It's a common question around the world and one we grapple with often in New Zealand. The method here was coupled nonlinear differential equations of cat, rat and seabird population dynamics. The result was feral cats had the greatest impact on seabird populations and urgently required management, and the very clear outcome, which I cannot emphasize enough, is that rat presence should never be used as an excuse not to manage cat populations. Cats do not control rat populations, ever. So this is Reunion Island, and if you want to know the exact field spot I was working on, it's that very high cliff on the top right-hand side of the island. Although the seabirds did gather at about 5 p.m. around the coast where all the bars were, and we could watch them while we sipped our French cocktails, unfortunately at about 5 p.m. they all fly to the highest summits on the island, which are about a 6-hour hike, where you then access your field site to study these birds. What we did was we created these partial differential equation models having a population of cats, a population of rats, and an age-structured population of seabirds. They have a juvenile phase, much like our grey-faced petrel in New Zealand. They go through 5 years being a juvenile at sea, and then they come back as adults and breed. So there's a lot going on here. The rats predominantly prey upon the chicks, and that's where the strongest impact is. The cats don't really prey upon the chicks because the chicks are safely locked away in burrows, but the cats do prey upon the adults. And the cats also prey upon the rats. The cats also prey upon other foods such as native reptiles. The rats also feed on other vegetation, and the change we can make in the population is removing rats or removing cats from the population. So putting all this together with a number of parameters, we could look at what the impact of cats and rats relatively were. And the take-home message really is that one adult seabird is worth about 5 seabird chicks, which is worth about 10 seabird eggs. So for every adult seabird you lose, you've lost the equivalent of 5 chicks or 10 eggs. So the adults are really most valuable in this population as the breeding unit. So there's 4 graphics here, and they all start off from the same place, which is that there's seabirds and rats on this island. The seabirds are slowly going extinct, or quite rapidly going extinct, in fact, and cats have made their way up into the mountaintops and are starting to impact on the seabird population. At the top, we have what's called bottom-up regulation. The rat and cat population, or the rat population in particular, is controlled by the food resources. Whereas down the bottom, food resources aren't so important, and what's important is how much predation on the rats there are from cats. On the left-hand side, we'll only be looking at cat control, whereas on the right-hand side, we'll be looking at much more labour-intensive cat eradication. So on the left-hand side, it's bottom-up, the rats are limited by food, and we're just going to control the cat population. So we do take about half of the cats out of the system. The rat population doesn't really change that much because they're still limited by food. They're happy not to be being eaten, but there's no more food, so they can't increase their population size. The seabirds still go extinct. Now let's try eradicating the cats from this bottom-up system. We eradicate the cats. The rats, once again, they're very happy not to be being eaten, but there's no more food, so they can't increase their population size. The seabird population makes a slow, painfully slow recovery, at least thankful that it's not being eaten by cats anymore. So now let's go to the top-down system. Here, the rats are being controlled by the cat population. So we control 50% of the cats. The rat population does increase in numbers because they are being released from predation, but the seabird population still goes extinct. Although the rats are impacting more seabird eggs and chicks, the cats are still there eating those very valuable adults. So let's explore cat eradication in this top-down system. We completely eradicate cats from this system, and our worst fears are realized, and the rat population explodes in numbers. The cats were controlling the rats, but even then, because the cats were having that strongest impact by taking out those valuable adult units, the seabird population now actually gets saved because it's the cats that needed to be removed from the system, and they're just as happy coexisting with rats in that population. So the take-home message there is that we should always really be managing feral cats because there isn't a situation where they're actually managing rat numbers. So it seems important that we actually need to test that hypothesis now. What does regulate rodent populations on islands? So the method here was community interaction modelling. The result, indeed, introduced mammals are bottom-up limited on islands. It's food pulses, it's a mast seeding event, it's those rainfall events on the dry islands, it's the seasonal summers coming along in temperate islands that feeds the ecosystem from the bottom up like a trophic fountain. And so you get more plant growth, you get more invertebrates, you get more mice, you get more rats, you get more stoats. It's exactly what we see in New Zealand with mast seeding. That's what controls the population here. But those introduced mammals then exert a strong top-down impact by preying upon all the native birds in the system. So the outcome of this study was that the removal order and the indirect effects of introduced mammals during eradication were determined, so we start to know what if you only take out these species from the system, what are the indirect effects, and what will be any negative repercussions that we have to prepare ourselves for. So to give you an idea, these islands all around the world start off in this native state with bird colonies, herbivores and primary consumers, native vegetation, below ground organisms, lots going on in the soil system. We come along as humans, we introduce mesopredators such as rodents, superpredators such as cats, herbivores such as rabbits, introduced vegetation. These all get into complex links with one another, which can make it very hard to unpack these ecosystems. So we do need to know the order in which we remove them. And this then spurred on further research by myself to look at things such as, well, what are humans' views when you lay those on top of this? What are humans' attitudes to rats versus humans' attitudes to cats versus humans' attitudes to rabbits versus what's your weed versus your prize flower in your garden. And then furthermore, working on these coral atolls around the tropics, I also realized there was something else going to happen in the next 50 years or so, which might render a lot of this work irrelevant. So I started to take into account what role humans have and climate change in the conservation of islands. And when we put this together on Europa Island, we got to see a picture here of cats exerting that predation pressure on the rats, and then the rats change their density. That affects their breeding, their home range size. Rat density is moderated by habitat quality and food pulses. The climate's driving the rainfall for those food pulses. It all feeds back into the system, and we get a relative understanding of top-down versus bottom-up effects, and when each of those is dominant in the system. So to sum that up, we know now that on these islands, the only resource input they have is from the marine interface. It comes from seabirds such as boobies and gannets, bringing food and marine nutrients from the sea to these islands. Those islands would naturally exist in a bottom-up limited state, but with animal and plant invasion, we have these animals such as cats and rats exerting a strong top-down limitation, and the invasive plants disrupt the bottom-up flow and redirect nutrients in the system in a way that completely disrupts ecosystem processes. So that's how we take a whole of ecosystem approach to conservation and don't just focus on particular species. So coming back to New Zealand now and tired of the warm tropics, off to Antipodes Island I go for a season. And here, the question became, what impacts do mice have on seabirds? We've worried a lot about rodents and rats in particular, but what are mice doing? And this was around the time when we'd started to discover on Gough Island that mice were becoming gigantic and attacking albatross that were 50 times their body size, very concerning. And there was a legitimate concern that that might be happening on Antipodes Island. The way to understand mouse diet without catching a lot of mice and dissecting them and hoping to get the one that had happened to get lucky and eat an albatross the night before was to undertake stable isotope analysis of mouse diet. The result was mouse diet seasonally shifted from land birds and invertebrates in summer to seabirds and plants in winter. And from this, we could report back to the Department of Conservation robust predictions of biodiversity outcomes from the mouse eradication. If you do mouse eradication, these are the species you expect will come back in the ecosystem. So here you can see the stable isotopes isoscape for summer and winter, and those circles there are the different individual mice and their samples, and we can see that in summer those mice are tending to dominate on those land birds and the invertebrates. Once again, just that resource pulse that comes on these cooler islands over summer. The tussock seeds, the invertebrates increase in numbers, the land birds start breeding, the mice switch onto them and prey upon them. In contrast, in winter, that resource pulse is finished. The mice are hungry. They have to scavenge on the remains of seabirds from the previous summer, and they switch on to plants as a diet staple is the only thing that still remains on the island over those cool subantarctic winters. And that's reflected here in the proportion that those different diet components are in summer versus winter. So the good news from this was in 2016, the Department of Conservation had the $1 Million Mouse Program and eradicated 200,000 mice at a cost of about $5 a mouse. And Antipodes Island is now mouse-free, and we've seen these results of this ecosystem booming once again with the invertebrates and the land birds recovering on it. So the question became after Antipodes Island, you've got a 2,000 hectare island that had 200,000 mice. You want to be quite confident you've got all of them, because the difference between no mice and one mouse can be quite substantial going forward. So can we calculate the probability of successful rodent eradication? The method here is a simulation model of the probability of successful eradication given no detections. It's the classic false absence problem in statistics. If I don't catch anything, is that because I'm really good at catching things and it's not here, or I'm really bad at catching things and there's a lot of them here? So we need to create a statistical model to balance the two of those out, so you can say with confidence when all of these repeated absences of captures are probably because there's nothing there. The result from this was we actually found biosecurity dogs significantly increase success confidence during a rodent incursion. And the outcome of that is the Department of Conservation now uses rodent detection dogs as a standard operating procedure for incursion response. And we had a real-time opportunity to do this on Great Mercury Island in 2017. So Great Mercury Island is 1,800 hectares. It's one of the top 10 predator-free islands in New Zealand. And this is the grid of traps around the island to monitor for rat reinvasion. You can see some patterns here. It's because there's coastal access points and roads on the island. But on top of this, we can have these traps. We can also overlay these tracking tunnels, a different type of device. You might have a rat with a different personality that prefers to go into a different type of device. So we don't just want to put all our eggs in one pest control basket. So we also have devices of a different type, but once again, a lot of them are occurring in the same place because of those accessibility issues of tracks in coastal areas. But we had some additional information on Great Mercury. They'd known they'd detected the rat down the southwestern area of the island. So if we delimited a radius of 2 kilometres around this area and we put out more devices to try and catch any rats in that area, so we intensified our effort there. And then furthermore, we took these rodent biosecurity detection dogs and we walked them around the island, and in particular we took them off track so they could cover some of the areas where we haven't necessarily got devices placed. The outcome of this is we can see that whether you're using a single device type or a multiple device type, so one trap or a trap in a tracking tunnel with different statistical probabilities, doesn't actually matter too much for your confidence of achieving rat eradication. And that's because although the difference in probability of capture might be 5% versus 10% per night, if you're leaving these devices out for 30 nights at a time, it comes out in the wash and they asymptotically basically have the same probability of detecting a rat. What becomes limiting here is where those devices are and whether you have 10%, 20%, 30% or 50% coverage of the island. In contrast, if we can delimit the incursion area and take a dog around it, that dog can go places where it can detect sign that is outside the area of our static devices that don't move around. A dog is essentially just a mobile trapping device, a sniffer trap that you move around on a leash. And you can see that big spike there from using the dog is because a dog may not walk through where the rat is, but if a dog walks through that scent area where a rat is, it has a guaranteed 100% detection of sniffing that rat, so you know that rat's there and can launch your incursion response there. So Doc learnt from this that taking these dogs out and using them strategically in the incursion response is a very, very powerful way to increase our ability to detect these reinvading rats. So as we started to undertake these eradications on these larger and larger islands such as Great Mercury, the question then became, do inhabited island human communities differ in their attitudes towards rat eradication? So the method here, which I worked on in collaboration with the Department of Conservation and my colleague Joe Ailey, who now works for the Department of Conservation, was surveys of attitudes to pest eradication. And the result was we found marked differences in community attitudes to pest management and rat eradication. Perhaps shouldn't have been as surprising to a biologist as it is, but every island we know has a different ecosystem. We know they have different mammals on them, so we don't go with a one-size-fits-all eradication method to different islands. So equally, when we want to engage with these inhabited island communities, communities, we shouldn't take a one-size-fits-all. These islands have different values, different things that motivate them, so what works to convince one island to do eradication might not be what works on the human community for another. So the outcome now, today, is we can tailor our social science tools to the target island. The study we undertook here was on 4 inhabited islands in the Hauraki Gulf. You can see the noises there where I started small on 10 hectares in the middle, but nearby Rakino Island had Norway rats on it and a community of about 20 people. Kowau Island to the north there has a community of about 200 people and has a good menagerie there thanks to Governor Gray of possums, cats, rats, stoats and friendly wallabies. Great Barrier Island has rodents and cats, and we have Waiheke Island with rodents, cats and stoats as well. Very interestingly, from a natural experiment perspective, was that the rats had actually been eradicated from Rakino Island. So not only could we look at the attitudes of these inhabited island communities to pest control, we could see how they changed if they knew the benefits of rat eradication, having lived through them themselves. So Joe's master's work found we undertook a pest management attitude scale where we saw what the propensity of the people living on these islands to support pest control was. On Rakino Island, we see independent of whether they're engaged in pest control themselves, there's a very high support for pest control and biosecurity. Similarly, we saw that on Kowau Island, but it dropped off very rapidly on Great Barrier Island, Waiheke, and our control comparison of the Auckland mainland. We can see that Waiheke and Great Barrier are really behaving more like the mainland, where if people don't engage in pest control themselves, they're quite removed from it and don't necessarily feel the motivation for doing pest control. And so this translates through to support for rat eradication. This is the proportion of respondents on each island that support rat eradication, do not support it, or are unsure. And although there's only actually a small amount of, often very vocal, but nonetheless small number of detractors on each of these islands, the number that are unsure of rat eradication, where doubt has been manufactured, is a lot larger on sites such as Great Barrier Island and Waiheke Island, where they're not necessarily so engaged in pest control. They haven't seen the costs of pests on their island or the benefits that follow from it. So then I started to wonder, do rat populations in New Zealand have different origins? What if we did a genetic survey, a phylogeographic survey of rats across all of New Zealand and understood the genetic tapestry of rats and what that might unlock for future potential control tools? So we undertook a mitochondrial DNA survey of introduced European rats, the ship rat and the Norway rat, across all of New Zealand. Hundreds of samples collected variously from island eradication campaigns, museum specimens. It turns out roadkill is a very good way to get rat samples from places you haven't got them before, or just cold door knocking on dock offices and saying, I'm coming through, have you got any dead rats in the freezer? What we found were there are at least 3 Norway rat introductions to New Zealand and 4 ship rat introductions. So the outcome from this is we could identify at a very high level what some of the barriers to rat invasion were when rats first arrived in New Zealand. So looking at the Norway rat here, we can see 2 invasions. There was the initial invasion to the North Island of that purple rat genotype, and that was also the one that ended up on our outlying islands, the Kermadecs, the Chathams, and Campbell Island. But in the south of New Zealand, we actually have a different type that arrived later from an independent invasion. They didn't cross over from the North Island, they came on an independent vessel. And those are the ones that also ended up on Stewart Island, but also some of the North Island ones or from the outlying islands have ended up on Stewart Island as well. And most interestingly, there's a very recent new genetic type of Norway rat found around Bluff area and making incursions inland from there, which suggests a more recent invasion through the shipping port of Bluff. Ship rats tell an even more complicated story. We have at least 4 types. I suspect with the recent SNP analysis we've done, we'll find that these are different independent invasions, but there's at least an orange type, a red type, a green type, and a blue type. So the red type is found throughout the southern North Island and then hits a reasonably hard barrier around Auckland and the Auckland Isthmus, and then the upper North Island has a different genotype. And then in the South Island, we see that same upper North Island type, except in the southwest of Fiordland, and around Bluff once again, where we see that, that red genotype. When we go to our largest offshore islands of Great Barrier and Stewart Island, Great Barrier has its own unique green haplotype, but that's been interbred with rat incursions repeating over time from Auckland of both the red and the orange type. And then if we head down to Stewart Island, interestingly, that has its very own unique genotype. But we also found that one of the landmark events that spurred our attitudes to rodent eradication in New Zealand was the invasion of Big South Cape in the early 1960s. Rats arrived on that island for the first time, and within 2 years, 3 species, 2 birds and 1 bat, went globally extinct, never to be seen again because of rats arriving on that island. Those rats that arrived on Big South Cape were the orange phenotype that comes from the South Island, probably from the Fiordland area as hypothesized from where the fishing boats were coming from, and had nothing to do with Stewart Island. So for the first time a few years ago, we ascertained where that rat invasion that was critical to New Zealand conservation came from. So one of the questions that I often have to grapple with is people say, well, invasive species are increasing biodiversity, and indeed New Zealand has more species than ever before, so if we're simply counting species alone, then we're doing really well for conservation in New Zealand. The method here is quite simply a thought experiment. And the result was we found alpha diversity increases from invasive species. Indeed, if you find any one island and chuck 20 or 50 or 100 new species on it, you've increased species richness, but it's the same species being introduced everywhere around the world and different ones going extinct. We're losing global or gamma biodiversity, and we're also losing the difference between islands. So 2 islands that had different faunas and floras become homogenized. So it's that global homogenization. And the outcome of this is a shift away from treating biodiversity as only being about species richness. So as a trivial example here, we've got 4 islands. They have a total down there at the bottom of 26 species native to them, each only found on that island. 5 introduced species are introduced to this archipelago, 3 on the second island and 5 on the third, 3 on the fourth island. We lose 3 species, 1 and 1, so we've had 5 extinctions. So in total, we still have 26 species in this archipelago. So you might think biodiversity has been conserved, but actually we've lost 5 species uniquely from the world and haven't gained any more because it's species that already existed somewhere else. So some of the methods we might use to restore these islands, on the first one and the fourth one, we might protect the first one as a pristine island that has no introduced species. That fourth one only has 3 introduced species. We might eradicate them and restore the island. So undisturbed natural habitat remains only in small pockets. Restoration efforts are successful on those small uninhabited offshore islands through eradications, plantings, and reintroductions of lost species. The second island has 3 introduced species, and it might have some novel ecosystems. So as a result of former land use, novel ecosystems develop. You get mixed species compositions of native and non-native species, and decisions around management of these novel ecosystems include a large values component about who values the native species versus the role of those introduced or exotic species, whether they're seen as positive or negative to the ecosystem. The final island is the third one. This might be more of a cultural landscape. Native biodiversity persists across the cultural landscape. Subtle changes in the management and behaviour promote it, but we've got to build this upon local knowledge, culture, and sovereignty of that island or those islands. And here we see the full picture of biodiversity. It's not just about species. There's genetic biodiversity, species biodiversity, functional biodiversity, and habitat biodiversity. And in the bottom there, you can see how species biodiversity is structured into that alpha biodiversity on the island, the beta biodiversity, which is the differences among islands, and the gamma biodiversity, which is the total biodiversity across all islands. So finally, I come around to philosophy and ask questions such as how do we balance the welfare of individuals, individuals' rights not to suffer or endure pain against higher levels of ecological organisation, such as the populations and species that conservation biology is trying to conserve. The method here is a typical philosophical one of thinking and debating, and the result was an argument for one-off lethal control of an introduced population to save a population or species would be a valid approach in conservation. And the outcome is we have a moral position stated for introduced predator eradication on islands. So here, this brings me to predator-free New Zealand, where I've focused my efforts for the last 5 years. In 1963, when Big South Cape Island was invaded, 0.5% of our offshore island area was predator-free. If you fast forward 50 years, we've eradicated rats from 1/3 of the islands of New Zealand, but only the small islands. 10% of island area is predator-free in 2014. Still something to be quite proud of. But there's still a lot more we could do on our islands. 50% of island area could be predator-free by 2030 if we eradicated predators from these large inhabited islands of Aotea, Great Barrier Island, and Rakiura, Stewart Island. And then if we look to the mainland, we see that 45% of New Zealand is actually currently under some form of predator management, not just for biodiversity, but for agricultural practice. A lot of areas where possums, mustelids, and rodents are being monitored and controlled to some degree. But how many of those sites have predator control to that gold standard of island eradication? The answer is in our 50 eco-sanctuaries, but they make up only 0.25% of the mainland area of New Zealand. So there's a lot more we could do to restore New Zealand to its former glory as an island seabird bird capital of the world. So I think on this journey, I've still got many more tools to find in my Swiss Army knife. The full conservation Swiss Army knife, I think, will be a very big and cumbersome tool like this, which requires not just myself, but the many others that come after me to find their Swiss Army knife tools and to use those. I want to thank the many, many groups that have funded me to enable me to achieve this research. I especially want to thank the many, many, many people that I've had the pleasure of collaborating with, and I am absolutely blessed, to be honest, that none of them has ever been anything more than a joy to work with and created amazing results that have helped continue to jettison New Zealand to greater conservation achievements. I want to thank my whānau, who are absolutely motivating and joyful to live with and be with, and to share my conservation with, but to provide a break from it as well. I'd like to dedicate this talk to my father, who sadly passed away 6 months ago tonight, who was a great man and just did his best to support me and share in me that enthusiasm for working with birds. So thank you very much. I'm happy to take any questions, and this is a website where you can learn more about the island conservation work we do at the University of Auckland. Thank you.